So I come now to the second part, and the second part is on the microbiota. So the microflora that is physically present mainly in the colon, partly also in the end of the small intestine. It's very important for the health of humans, and it's also an instrument, a tool that we can try to regulate in order to benefit the overall health of the host. In order to do that, you need to take into account three different potential mechanisms. The first one is the host itself, which is actually the human body. The second one is the food that we are eating. And the third one are the microorganisms themselves, because you have actually a sort of a host microbe interaction. And this is determined by a whole set of compounds, which you can see here. So for the host, this is the specificity of the adhesion and the absorption capacity. So it means that if you want a microorganism to be active on human metabolism, the first thing you need to do is to see that the microorganism gets attached to the wall of the intestine. If it's not attached, then after a couple of times, it will just be removed by the fecal material. So it will be lost. So in order to have physical impact, you need to get it absorbed. So to do that, you need to know the adhesion positions and you need to know the absorption capacity of the wall. The second is the rate of secretion and the composition of everything that is secreted from the intestine. This also determines to some extent the composition of the microbiota, because the mucus layers and all the different compounds that we are releasing from the host is host specific. So there are slight differences from person to person, and this could also change the overall composition of the microbiota. We can also influence the motility of the intestine, so the speed by which the food is transported through the GI tract, and then the frequency and the amount of the food or the feed that we take up. Yeah, so all these elements that are host related will determine the composition of the microbiota. The second option you have is the form, the composition, and the degree of contamination of your food. And do you eat it in small particles? Do you eat it in the solid form, in the liquid form? The composition of it, is it a carbohydrate-rich substrate? Is it rather a lipid-rich substrate? And are there microorganisms there that could potentially be pathogenic, so dangerous for you? So based on all these elements, again, the microbiota composition can be influenced. And then finally, we have the microorganisms themselves, which also need to have specific adhesion positions. Otherwise, they cannot interact with the adhesion positions of the host. Some of these bacteria need growth factors. A good example is bifidus, which is commercially available in the supermarket. You can buy these types of products that are enriched in bifidus. But the bifidus bacteria is an anaerobic bacteria. So strictly anaerobic, it can only grow and develop in anaerobic conditions, so without oxygen. So in order to cultivate a bifidus on an industrial scale, you need to have these bioreactors that are completely filled with nitrogen and that are free of oxygen. And you need to add additional growth factors to selectively stimulate the growth of bifidus. And this is very expensive. Yeah? So bifidus today is a first generation probiotic bacterium, which is now more and more replaced by the second generation bacteria, which are mainly lactobacilli. And lactobacillus is more easy to cultivate. It needs less oxygen. It doesn't need these complex growth factors. So it's easier and cheaper to produce it compared to the bifidus factor bacteria. But both of them, bifidus as well as lactobacillus, are healthy bacteria. So we need to have enough of them in order to have a healthy microbiota. And some enzymes that are also produced by the microorganisms could play a role in determining the composition. So if you take all these elements into account, then you will create a so-called microbial system that can quite easily adapt and that can mutate. And this microbiota system is different for every individual. And it can very quickly destabilize. If you go to another country, you eat a food that you're not used to, it can be that your microbiota is not stable and you get diarrhea, which we call traveler's diarrhea. It's very temporary. If you are there for a couple of weeks, it will stabilize again and you don't have any problems anymore. But the system can rapidly change, but also rapidly can destabilize, depending on the outside environment. Here you can see the adhesion positions, which is a very important field of research in human nutrition today. So you have here all the different epithelial cells present and residing at the gut wall. You get all these specific receptors that are host specific. And then you get bacteria that have the same receptors, so they are capable of binding. This one can bind, but this one cannot, because these arms here, which we call the vimbrae or cell ligands, are actually not fitting into these receptors. So as a result of that, it will never attach, and it will just be flushed out the next time there is a removal of fecal material from the host. The second thing is the same. So here you can have an interaction because there's a perfect fit between the receptors and the different vimbrae. But it could also be that viruses or toxins have the same type of ligands and that they can also interact. So there is a competition between the beneficial bacteria, the viruses, and the toxins to interact with this interficial material. And then the question is which one can survive, which one is dominating. Well, you always hope that the positive bacteria can dominate and that we can remove the viruses and the toxins. There are different types of bacteria inside the colon. I think we have about 10 to the power 14 bacteria, and there are about 10 to the power 13 eukaryotic cells in the human body. So it means that we have more microorganisms inside our cells than that we have human cells. We are a living bioreactor, a walking bioreactor with microorganisms that definitely can influence the health of the host. You see potential negative organisms that are negatively influencing health, and you see positive organisms that positively influence health. The positive ones are lactobacilli and bifidus. Bifidobacteria, the first generation, lactobacillus, the second generation bacteria. So these ones we want, and we can all find it already back in different types of commercialized products. While the other ones, like for example the streptococci, the E. coli, the enterococci, the clostridia, so all these types of bacteria probably are a little bit more negative. So we do not want to have them in too dominating amounts. So you always have to find the balance between the two with the hope that we have the positive bacteria dominating over the negative ones. These ones are positive because they can produce acids. And with these acids, they can actually lower down or slow down the production of the pathogens. And they are producing a lot of useful substrates and metabolites that can be absorbed in the human body and that can have physiological relevance. So how is microbial metabolism taking place in the colon? It's a quite complex process because you have all these bacteria that are all interacting with each other, and one bacterium is breaking down a substrate, the other one takes it over and then further metabolizes the substrate. So there's a lot of cross-feeding and a lot of interaction between these bacteria, which makes the process quite complex. If we look at it from a biochemical way, then the first thing we can say is that the basic substrate for all these bacteria are hexoses. And a hexose is actually a monomer mainly of sugars, for example, glucose or other derived components. And once they make it to the colon, they are the ideal substrate for the bacteria to grow and to survive. So they go through the different biochemical pathways, and independent of what actually all happens intermediately, there are a couple of end products that will accumulate in the environment. The first one is acetate, the second one butyrate, and the third one propionate. So these are the so-called short-chain fatty acids. Fatty acids with a limited number of carbons. Butyrate has 4 carbons, propionate 3, and acetate 2 carbons. These ones are directly usable and metabolizable by the host. So the human cells are using butyrate near the intestinal lining. So the epithelial cells near the gut epithelium will mainly start using butyrate. The propionate goes inside the body and can actually act in the metabolism, for example, in the liver. And the acetate is an energy supplier. So in case you need to get some extra energy, you can actually see that more acetate is absorbed because this one is supplying energy to the human body. The lactates, so milk, so lactic acid, which is directly produced from pyruvate, is only produced in exceptional conditions. For example, if I have a potato, and you can peel the potato and you can just eat it raw. Just imagine you're not going to cook it, but you're eating it raw. In those conditions, the starch inside the potato is not that well digestible. It can go to the colon, and all the bacteria will start using it. And then you get a lot of lactic acid production, especially because these bacteria are working in an anaerobic way with the glycolysis, and then you get a lot of lactate formation. You acidify the medium, and this destabilizes the flora, and you get diarrhea. So if people eat, let's say, 3 or 4 potatoes that are raw, you will probably get diarrhea because there's an overload in the colon with an acid production, and this will normally lead to destabilization. In normal conditions, the lactate production is lower. It's mainly the fatty acids that are produced. Second one is ethanol, which is also not that much physically there in humans. There may be ethanol production in some conditions, but I think generally speaking, this pathway is not dominating compared to the free fatty acids pathways. And then a last pathway that you have here on the top is the production of methane gas. That's quite important because methane gas is actually contributing to climate change. As you probably know, that's why we have these ruminant animals that need to be a little bit limited. Because if we have a lot of animal production with a lot of cows, for example, there's a lot of methane, which is actually released into the atmosphere, and that can also lead to increase of the Earth's temperature, so climate change conditions. There are some individuals that are producing methane gas. Some other individuals will not. So it all depends a little bit on which type of microbiota that you have. So if we look a little bit to the microbial processes that take place in the different GI tract models, then we first look at the substrates. So the substrate we have is enzyme-resistant starch. So it means that there is part of the starch which normally should be digestible in humans. It's an alpha-1,4 bond. So in principle, it should be broken down. But in special conditions, I already gave you one example, the raw potato. The raw potato contains starch, but the starch is not accessible. So it's physically entrapped in the middle part of the cell, and the enzymes can't reach it. In those conditions, the enzyme or the starch will go to the colon, the bacteria will start using it, you get acidification, and you get destabilization of the colon flora. So this is a perfect example of a fiber, provided that the starch is enzyme resistant. A second example are non-starch polysaccharides. So these are the most well-known fiber components, like for example cellulose or hemicellulose or pectins. So these are all cellular walls that can actually make it to the colon, and they are the ideal substrate for the bacteria to grow. And then we have some special cases, lactose, for the lactose intolerant people, where lactose comes in, it is not digested, it goes to the colon. So in the colon, you also get lactose fermentation. Sugar alcohols, these are derived from sugars. For example, lactose will be turned into lactitol. Maltose will be turned into maltitol. So all types of sugar alcohols with an alcoholic function at the end. These ones are low caloric and they don't acidify the mouth, so they will not have an effect on tooth caries. And they are low calorie, so they give you less energy. So these sugar alcohols are also used quite a lot in light products, for example, in light jams. And so if you buy jams to eat and they are light, just check it. There's quite some sugar alcohols there. The disadvantage of a sugar alcohol is that when it reaches the colon, the bacteria start using it and you get a lot of gas formation, flatulence, which of course, is not that comfortable, and it can also, in worst case conditions, destabilize the flora, and then you get diarrhea. And some people are quite sensitive for that, some other ones are not. And then the final one are the oligosaccharides. A good example are when you're eating beans, legumes, and so they contain compounds which are called oligosaccharides, short chain sugars, that can actually create flatulence in the colon. So they're not digestible, they go to the colon, bacteria start using them, producing gases, and then you also get flatulence. So all these substrates will enter in the colon. You get an anaerobic microbial fermentation. Anaerobic means no oxygen, and you get a lot of end products. And the first one are the bread and the flatus, where volatile components can be removed, like hydrogen gas, CO2, and the methane gas. And so this can just be expired or it can leave the body by the flatus. The second one is acetic acid, propionic acid, and butyric acid. These ones are physically absorbed at the level of the intestine, and they can create energy for humans. And they can be used for energy recuperation, or they can influence human metabolism. So you can find them back in the blood, you can find them in the fecal material. And then the third reaction we have is production of microbial biomass, for which we need nitrogen. And that's quite important because if there's enough supply of carbohydrate, then the microorganisms will start using carbohydrate. And the nitrogen, which is physically there and coming from proteins, will then be used to produce microbial biomass. So if there's enough energy coming from carbohydrates, then the microorganisms can multiply and they can grow, they can get bigger, so you get more fecal material, more microbial biomass. And in order to grow, you also need nitrogen, because you need to have amino acids that can actually build microbial protein. So in this way, this is the most healthy way of working. A lot of energy coming from the carbohydrates and the nitrogen completely turned into the microbial biomass, which goes to the fecal material. And in this way, you can produce quite a lot of fecal material. For example, if you're eating a lot of dietary fiber, you definitely will have a larger volume of fecal material produced. If you don't eat these types of fibers, then the volume of the fecal material will physically be reduced. The driving force of the fermentation reaction is hydrogen gas. And you can see the hydrogen gas here in the middle. It can actually lead to production of acetic acid, which will be used for energy. You could lose some hydrogen gas itself. This is not efficient. I mean, the microbial system will try to avoid that because hydrogen gas is important for all the physiological reactions. So the more hydrogen gas you physically remove or lose, the lower the efficiency of the process. So let's say that an average microbial process in the colon will have an efficiency degree of about 90%. So it means that 90% of all the hydrogen gas will be entrapped and will be used in all the other reactions. 10% of the hydrogen gas may either be lost or it may be used by other reactions that are not here on the slide. So this is what we actually call efficiency, the way by which hydrogen gas is recovered and used to produce useful metabolites. You can produce the biomass, which we already discussed with the nitrogen. You can also produce methane, and you can produce, in some conditions, hydrogen sulfides. The hydrogen sulfides you can recognize easily because they have this very strange smell. It's a little bit the smell of rotten eggs, which is a sulfated compound that comes into the air and that is very easily found back. So if you have a lot of sulfate producers or consumers here in your colon, then you might be also an H2S producer. It's not the same for everybody. So some of them are methane producers, some of them are H2S producers. So overall speaking, the discussion is still going on in the literature, but generally speaking, we can assume that methane as well as H2S are not that positive for human health. So in general conditions, what we try to do is to limit a little bit the methane production and the H2S production and to maximize the production of the free fatty acids because they are in most conditions quite useful for you. The second type of reaction that we can have is the effect on protein. So you could break down carbohydrates, which is the preferred substrate, but you can also break down proteins. And when a protein is not well digestible, it will pass the small intestine, it will go to the colon, and the bacteria can start using it, especially for plant-based protein, which is entrapped by different types of fibers. It may be that the digestibility is lower compared to an animal-based protein, which generally speaking is quite well digestible. And it can go to the colon where the protein will be used by the bacteria. So if we have microbial metabolism of protein, you can have again the formation of hydrogen gas and CO2. They can go to the bread and the flatus. You can have the free fatty acids in the same way, go to the blood and the fecal material. And you have the biomass, which can go to the fecal material. But in addition to that, there are two routes which are a little bit specific for proteins and peptides. And you will not find them with the carbohydrates, but you can mainly find it for proteins and peptides. So what happens here is production of amino groups, amines and phenols, and these are potentially not so positive for health. So when they are produced in the colon, they can interact with the colon wall, they can induce irritation of the colon wall, which can potentially also lead to negative effects like colon cancer. So we do not want to have too much of these components produced from protein in the GI tract. You can also produce branched-chain fatty acids. They are produced from branched-chain amino acids like isoleucine, for example. So the amino group is removed and the remaining acid is called a branched-chain acid, can actually go to the host, and from the blood it can actually be brought to the adipose tissue where it can be stored. So if you take a biopsy of the adipose tissue and you see a lot of branched-chain fatty acids, it actually means that the colon flora is physically producing branched-chain fatty acids from proteins and peptides. Yeah, so you get mainly amino acid metabolism at the level of the colon. And this brings us to a little bit the physical process itself. So here you can see a slide with how the large bowel or the colon looks like in humans. So in the middle section, we usually have the small intestine, which is enrolled here in the middle of the belly. And then you can go into the ascending colon, which is the first part of the large bowel. And this one can go up from the belly until you meet the midriff. And there you have the transfer colon in the middle. And then it goes into the descending colon at the end, ending in the end here with the rectum. So you can already see that you have different compartments, let's say, an upward compartment or horizontal compartment and a lower compartment or a descending compartment. And in all these compartments, the activity of the microbiota is different. So you can already start thinking a little bit on what happens in the ascending colon, what can happen in the descending colon. And this brings me automatically to my second question for problem-based learning, which you can actually see here. I will come back to the figure immediately, but this is a question for all the groups B, where you can think a little bit about a microbial fermentation process and how this could influence the overall health of humans. So the question is the following. Colon cancer is mainly found in the last part of the large intestine. So it's mainly found in the descending part of the colon. Explain this phenomenon, taking into account that carbohydrates always receive priority as energy supplier compared to proteins. And the metabolic reason for that difference between carbohydrates and proteins will further be explained in chapter 5 of the course notes. So you might already check a little bit there if you want to know better some basic mechanisms by which carbohydrates and proteins interact during energy metabolism. So that's the situation. So I'm going back now to the slides that I showed you here. Belgium is a quite unique country. Well, not unique, but in the sense that in Flanders, so the region where you are staying now, in that region, we have a Flemish program running. So it's actually sponsored by the Flemish government, by which all individuals, all residents of Flanders, with an age higher than 52, receive in their mailbox an invitation to have a screening for colon cancer. And this is completely paid by the government. So it's a free service that is supplied. So what they actually do is they send you an envelope. It comes in by the mailbox. In that envelope, you will find a small box, which is a plastic box. And you have a piece of paper, which is a special absorptive paper that you have to put into the toilet. And so this one will collect the fecal material. And then inside the plastic box, there's a small needle. You take it out and you just wrap it on the surface of the fecal material. It's only a very small amount of fecal material that you will recover from that. But you put it back into the plastic box, you close it. It's an anaerobic and aseptic medium. So you close it, you put it back into the envelope, you close the envelope, and on the envelope, the address of the lab is already indicated. So you bring it back to the mailbox, you send it, it goes to the lab, they analyze this fecal material for you, and this is the blood inclusion assay. So it means that in the lab, they will check whether your fecal material contains too high amounts of blood cells, red blood cells. If that is the case, then they will send you an invitation to come in for a colonoscopy. So it means that they will check a little bit the inside of the colon to see if it's still intact. If the test is negative, they will not invite you. And they will repeat the question to you every 2 years. So if you're 52, you get the letter. At 54, you get it. At 56, etc. So each time you get a letter so that you have a preventive screening program to evaluate the health conditions in the colon. When you have to go for a colonoscopy, you come into the hospital. They ask you to be completely fasted, because if food is actually present in the colon, then they can see nothing. So you need to have a period of fasting, or in some conditions, they might ask you to take a product that can release all the particles of food from the bowel. Yeah, so it's actually a sort of a system to, let's say, clean the bowel so that it's completely empty for the colonoscopy. What they then do is they take a small piece of a sort of a needle, but it's a long thread. With on the top of that, there's a very small micro camera. And with that camera, they go into the rectum and they just follow a little bit here the condition of the wall. So they can go up and they can just look at the wall structure and see if you have some perturbations, which we actually call the tulips, which means that you have a sort of an extension that is physically present near the wall, and that could be the onset for tumor formation. If they see that, they will cut it away. Yeah, so if they are early enough, that's why they want to do the preventive screening. If they're early enough and they can cut it away, in principle, it's not dangerous anymore. If they leave it as it is, it will start growing further and further, and then you get a colon tumor. Now you can imagine that if you want to do a colonoscopy and you look at the structure of the colon, it's not easy, because you have to come in here, you have to go up to the end, then you have to bench it almost 90 degrees, and then go the whole way, and again bench it to go to the front. Yeah, that's a little bit complicated. So the real colonoscopy, as they do it, will mainly act on the last part of the colon. So only that part. And so they mainly do that there because the majority of the potential tumors that are found are mainly found here. Yeah, at this part in the descending colon. There may be tumor formation there, but it's more limited. And if it is there, it is probably also genetically determined to an important extent. So my question now is for the groups B, why do you think that the formation of the tumors is mainly done in the descending part of the colon and not in the ascending part? So try to find mechanistic explanations for that, and then you can immediately also see the benefit of just screening here in the descending part of the colon. Also, these answers in maximum 10 lines, please, and to be uploaded on the Ephora website by the deadline. I think last week I explained a little bit to you the use of organoids, and I can imagine that it might be a little bit complex for you, a little bit technical. But with this slide, I hope I can convince you the importance of that new technology. And on the internet at Ephora, on the references, you can find a paper that is dealing with the applications of organoids. And on the left side, you can already see what we discussed last week. So the fact that you have the crypts will be isolated so that you can actually seed them. You could separate cells if you want, and there are cell sorting devices available. You bring them into the right culture medium. These are the stem cells, and then it starts growing into a full particle, which we call an organoid. And under the microscope, it looks a little bit like this. So you see these lopped type structures. So this is typically for the new crypt cells and crypts formations that are taking place. Now, this organoid technology could in the future also be used to treat patients with colon cancer. And the idea is the following. So if you have a tumor, which occurs here in the colon wall, what they actually need to do is to remove the tumor. Yeah, so they have to cut away the tissues, and they also have to cut away a little bit healthy tissues on both sides of the tumor to be sure that everything is removed. But most of the time, when that happens today, these patients lose part of their colon, and in the worst case situation, they actually lose the whole colon, in which they become an ileostomy patient, which means that they will actually collect fecal material at the end of the small intestine. Yeah, so this is not a very easy condition to live. So of course, we hope that patients could maintain their colon activity as long as possible. In order to restore the situation, the idea is to start using organoids. So you cut away the part of the tumor, and then you take a stem cell, which is isolated from the healthy part of the colon. So, for example, from the ascending intestine, and you use that to produce new colon tissue that you actually bring into the system. So you bring in the new wall material that can regrow again, first in this way, then it forms the lub structures, and finally it may form a completely new wall. With the advantages of this system is that you are using stem cells from the same person. So it means that the immune system will not respond, will not actually deter it or destroy it, because it's actually cells coming from your own body. Yeah, so the chances of failure are in this case much lower. The technology is not yet optimized yet, so the organoid technology is still in full development, but the idea in the future is that it could be used to help colon patients to recover part of their colon wall. The last part of the presentation is about axenic and gnotobiotic animals. These are special animals, mainly rats and mice. They don't do it for bigger animals because it's too expensive, but you can do it for very small laboratory test animals like mice and rats. And what you actually do is you bring them into a sort of a reaction chamber, which is completely free of air. So it's in an environment where it's completely sterile. So there are no bacteria, whatever there. And all the food that comes in needs to be sterilized. So it's sterilized in these compartments here, these sterilizers. And then it is brought into here at this part by a special system that can also avoid contamination. So the inside of the vessel is in overpressure, so that you don't have air from outside coming in, because then you might get contamination. But it's in overpressure so that inside you can work actually with a sterile animal, which we call an axenic animal. So this is an animal that does not contain any microorganisms. And a gnotobiotic bacterium or a gnotobiotic animal is an animal that starts to be axenic, but then you add deliberately one individual type of bacterium, just to study the behavior of these bacteria in the absence of the other colon floras. Then we call it a gnotobiotic animal. These ones are quite important in the pharmaceutical industry. They use these animals quite a lot because they want to evaluate what the impact of the microbiota is on colon health. So if you remove the microbiota, you get a perfect animal that can just survive without the impact from the microbiota, and you can compare it with an ordinary animal that still has the microbiota operational. And in this way, you can see how important are the microbiota for us. So if we do these tests, we can look a little bit at the characteristics of these animals, and they seem to behave in a completely different way compared to the classical bacteria that do contain microbiota. For example, there's an effect on peristaltis. There is also a change in morphology, whereby, for example, the cecum in those animals is getting much bigger because there's a lot of water inside that cannot be removed. And the reason why it's not removed is because there's a very thick layer of mucus near the intestinal wall. And this mucus layer is normally removed and destroyed by the bacteria. But if there are no bacteria left, then the mucus will grow indeliberately, so without any interruption. And then you don't have any exchange of water anymore. And that's the reason why the cecum will blow up in volume. All types of enzymes are lost, so there's no lactic acid production, more urea and less protein formation, no branched chain fatty acids, bile acids are not modified, no vitamin production. So there's a lot of metabolism that is actually changed if the bacteria are gone. And in the animal production industry, in the past, especially now, is becoming more critical, but in the past, they have been using quite a lot of antibiotics in order to kill the bacteria and in order to have more efficient intestinal absorption, which was important for the rapid growth speed, for example, of pigs or cows or whatever. So they actually tried to limit, in this case, the potential impact of the microbiota. Today, microbiotics are used far less or even not used anymore because you could create antibiotic resistance, which is a quite dangerous situation also for humans. So this microbiota system we would like to optimize. So there's a whole industry that has now started with the production of probiotics. And a probiotic is a live microorganism that, when it is administered in sufficient quantities, can confer a health benefit on the host. And so it means that you give a bacterium, for example, through the food. You hope that it will survive until it enters in the colon, and there it will stimulate the growth of healthy bacteria. And so in this case, we call that a probiotic bacterium. The first generation probiotics are the bifidus. I already explained that to you. It's a very healthy bacterium, but to produce it industrially, it's more complicated. So that's why they switched to the second generation bacteria, like, for example, lactobacillus, which is not so difficult to cultivate in the industry. You can use a little bit of oxygen. You don't need these complex growth factors. So it's more efficient and cheaper to produce this one than that one. And there's already a next generation probiotics, like, for example, Akkermansia, which is now being associated with breakdown of mucus. It can also have several metabolic reactions in the human body. So these ones are new upcoming bacteria that might be used in the future for actively stimulating health in humans. It's very difficult to say which type of bacteria people need. Probably you need many of them, and you probably need a certain balance. The best way we can formulate it is that we need to have bacterial richness, which means that there must be a very broad range of bacterial diversities. So different types of species that are physically there and that can break down a very big set of different types of substrates. So the more diverse the microbiota is, potentially the more healthy it is for the human body. And I end with the last slide, which is this one. I deliberately want to use it. The article, I think, is also online. It's Jugendholz et al. From 2018. My basic message is that a lot of research today, also in terms of the microbiota, is actually done with mice, which is the standard testing animal, for example, for studying colon cancer. And there are a lot of cancer institutes that are using millions of dollars and euros to investigate potential colon cancer effects and how we can treat it. The basic model to study there is the mice. And there are many reasons for that, because there are many mice models available with tumors, where you can induce tumors, where you can also have different dietary regimes. So the medical sector uses the mouse mainly as the basic animal of research. However, if you look a little bit to the digestion process in humans, and you compare it with the digestion process in mice, you will see that there are several differences. And the mice is not an ideal example of a digestive process in humans. This can be seen from the morphology, so the size of the different organs, the residence time in the different compartments, even the tumor formation. For example, if you work with mice in tumor research, the tumors in the mice are mainly developed in the small intestine, while in humans, the tumors are mainly produced in the large intestine. So be very careful with the mice model. It's an interesting model, it's easy to use, but it's not always representative for the human situation. And there are intermediates. You could, for example, use pigs, but pigs are, of course, expensive. They need much more material. You could use mini pigs, which use less material, but the mini pig as such is bred in a special way and is still quite expensive to use. So we don't have a lot of alternatives, but strictly speaking, the mice is not an ideal model if we're talking about digestive studies. So with this, I think I finished everything I wanted to discuss with you on digestion. So I think I will stop the lecture here and the recording. I will stay an additional 10 minutes. If you have any questions for me about this lecture or about the rest of the course, you can always ask me. So I thank you for your attention from being here. See you back next time. Bye.